announce the talk time as 11:30. And some of you might have read the talk time as 11:45. But please have a little more patience. I think he is still in the VC's office in the university. So we're waiting for him to join us. So thank you for your patience. And yeah, chit chat, have fun till then. Yeah. welcome. We at the National Center for Cell Science, NCCS, are very pleased to host today's talk by Sir Richard Roberts in association with the Savitribai Phule Pune University. We will begin with a welcome address by Dr. Mohan Vani, Director, NCCS.
Very good morning to all of you. It is my great pleasure to welcome you all for today's Nobel Laureate lecture by Sir Dr. Richard Roberts. Very warm welcome to Dr. Roberts. Sir, we are really honored and appreciate the time you have given this morning for the staff, scientists, students, and public which we have invited from most of the colleges from Pune and Pune University. I also would like to thank all the distinguished guests, faculty, professors from Pune University, students, ladies and gentlemen. I thank you for joining us today to grace this occasion. Today's talk is jointly organized by National Center for Cell Science Pune and Savitribai Phule Pune University. Sir, National Center for Cell Science is a premier research institute of Department of Biotechnology, Government of India. NCCS is doing cutting edge basic and translational research in the areas of modern biology and medicine, including cell biology, cancer biology, metabolic and infectious diseases, then human microbiome, neurobiology, structural biology and bioinformatics, and also stem cell and regenerative medicine. We have more than 125 Ph.D. students on campus, and there are 35 independent research scientists. They have their independent research groups. Till date, NCCS, more than 350 Ph.D. students are graduated from NCCS, and I'm very proud to say that all our Ph.D. students are placed in well-reputed international and national institutes and universities. So I'll just give the brief introduction about Dr. Roberts, and Dr. Shekhar Mande will give the introduction in detail. Sir Dr. Richard Roberts, a Fellow of Royal Society, was awarded 1993 Nobel Prize in Physiology and Medicine for discovering a very important feature of genes. His discovery of introns in eukaryotic DNA and the mechanism of gene splicing was truly a paradigm shift. How split gene information is edited is very important to understand, not only for during the development but also in a health and disease condition. I am sure that everyone will enjoy the lecture of Dr. Roberts, and it will really motivate and encourage all our scientists, PhD students, the future PhD students to conduct the quality basic research to understand the biology and pathophysiology of human and animal diseases. So please join me in welcoming Dr. Roberts. Now I invite Dr. Shekhar Mande to introduce Dr. Roberts. Dr. Shekhar Mande is a former Director General CSIR and former Director of this institute, and currently he is a distinguished professor at the Center of Bioinformatics, Pune University, and honorary distinguished scientist at NCCS. Dr. Mande.
Thank you, Mohit. Friends, it's a great pleasure and privilege to say a few words about Professor Sir Richard Roberts to all of you. Richard's work, everybody knows him as Rich. His work is too well known, is there in all the textbooks and all, is taught all over the world in the biology textbooks, and perhaps the students may not even realize that the work was done by Rich. So we're going to hear from him about that already, what is taught to you in your curricula. For the non-biologists, I believe there are some people who are not from the biology background here. For the non-biologists, I mean, you know that each of our cells carries DNA into our cells, and part of the DNA, a very small part of the DNA, actually codes for information that is translated into proteins. I mean, this small part is about 2% of the DNA in human genome, what we call as genes. Now, that was known for a long time that there exist genes on the DNA. But what Rich started his work in 1970s, in mid 1970s or early 1970s, his work on adenoviruses then led to the discovery that the genes are not really contiguous. They also are fragmented into portions, some portions of which eventually go on making the proteins, the information of that goes on making protein, and some portion does not. What is now called in the textbooks as introns and exons. There's the discovery of Rich. So many of the biology students probably would have read this, and you also answer your questions in the examination about this, but it's the person who discovered that here. And as Mohan said, he was very richly awarded 1993 Nobel Prize for the same discovery. But a few other things about Rich I would like to mention here is that what we see today as the open access publishing movement around the world, in which there is an author pays model, that authors for publishing paper in journals, authors pay to the journal, and in turn journal makes the article available to everyone in the world free of cost, a downloadable article. And that open access movement, origins of which also Rich, when he was the editor-in-chief of nucleic acid research in the early 2000s, he pioneered really that movement around the world and has taken now firm roots all over the world, the open access movement where authors pay the journal and journal makes it available to everyone in the world. So firm that probably it's not to the liking of some of us now, especially in the era of what we call as publishing, which is not very desirable. So there are journals which have been making business out of these kind of things. But nonetheless, the movement, open access movement, was pioneered by Richard long ago. Richard also is a very strong supporter of computational biology and bioinformatics. When he was the editor-in-chief of nucleic acid research, he started a special section on databases and web servers and all. And many of the bioinformaticians around the world, including me, have benefited by this, and we have published our papers in nucleic acid research describing our databases and so on and so forth. The third and last thing that I would like to say is that he also is a strong proponent of the genetically modified crops, and I believe he is giving a talk tomorrow in Ferguson College on GMOs. And I was just telling him that only about a week ago, India did give approval to its first GM crop, that is the GM mustard, as all of you would have heard. The Genetic Engineering and Appraisal Committee, in short GEAC, has given approval for the cultivation of GM mustard, which actually you'll be very pleased to know about that, despite all the opposition in the last 30 years. So in summing up, some of you know that I have been with the University of Pune for the last 10 days, and I have an honorary position in this institute. So it's my special privilege to welcome Rich amongst all of us on behalf of both the Savitribai Phule Pune University and on behalf of National Center for Cell Science. Ladies and gentlemen, please give a big hand to Rich, and I invite him to speak.
Okay.
Before we begin the talk, we would like, Dr. Vani would like to welcome the guest with a bouquet.
Oh,
Do you want me to hold on to the cord?
no, I'll just put it down there. Thank you. Thanks. Now it's important that you tell me how long you want me to talk for, because once I get started, it's sometimes very difficult to stop me. So what is the time constraint here?
1 hour including question answer.
Okay. Okay, so what I want to do today is to take you through my life, how I sort of got educated in the first place, how I got interested in science, and then the kinds of things that I've done. But what I'm going to try and do during the talk is to give you some indications of things that have been very important for me, but which I think will also be very important for many of you students. I think there are lots of things that you don't fully realize. One of them is the part that luck plays in your life. And you will see, I will show you plenty of examples of this, and I'll talk about it some more at the end. But the other thing that so many students find is they fear failure. And failure is actually one of the best things that can happen to you. And when teachers or parents or someone else start criticizing you because you failed, it's very wrong. It really is very wrong. If you look at almost everybody who started a big company, they started a small company first and it failed. But they learned from that failure. They learned that you do a post-mortem, you find out why you fail, and you make sure you don't do the same thing again. And in science, this can be absolutely wonderful, as you will see. So let's get started here. Oh, what is... Did someone take this off presenter mode? I put it in presenter mode. No. No. No, I just had it in presenter mode. Did you do something? That's right, perfect, okay. So, I grew up, well, I was born in Derby. It's this little town here right in the middle of England. I was born in 1943, which was towards the end of the Second World War. And I was very lucky from day one, because I lived on a street where the German bombers were busy flying over to try and destroy the Rolls-Royce factory that was making all of the engines for the Royal Air Force. And they wiped out one side of the street, but it wasn't the side I was living on. So that was an incredible piece of luck. You know, 50-50 chance, two sides of the street, mine was okay. Now, when I was 4 years old, my parents moved to Bath, shown in the right-hand slide. It's exactly 100 miles due west of London. It's a fabulous city. I think if you ever go to England, you should make a point of going to visit. It's very easy from London. It's an hour on the train, and then you can be there and back. It's very nice for the day. But it was there that I really grew up. This is all of my childhood memories really come from Bath. See, we're having some problems here already. Okay.
Yeah.
Huh?
Just use this up
Yeah,
and down
I
key,
think-
probably that will work.
Yeah, this isn't working. Okay.
Yeah.
Okay, so something very important happened when I was about 8, 9 years old. I went to this school. It's a very small school. You used to walk to it from my house. And when I used to leave the school, the headmaster would often give me a slip of paper, and it would have on it a little mathematical puzzle. And I would take it home and do it. And as a result, I fell in love with mathematics. I thought this was just fantastic. And I also fell in love with puzzles and games and so on. And it never occurred to me at the time that he didn't do this to everybody. But for some reason, I was the only person that he was doing it to. And why? I've no idea. He must have seen something in me at even that early age. But this, for me, was really a striking point. I've loved mathematics ever since. So even today, I love it. And as you will see, I do mathematics, but not the kind that I did when I was a kid. So that was very important. Now, one of the other things that you have to realize, this is just giving you an example of how nice Bath is. On the bottom left are the Roman baths. Bath was built where there were hot springs, and the Romans built these beautiful baths. They're really quite amazing. And then on the right-hand side, you see some of the beautiful architecture. This dates back to the Georgian times. And the whole city it's just gorgeous, absolutely gorgeous. But when I was about 11, I read a book, and the book was entitled How I Became a Detective. And, you know, I read this book, and by the time I'd finished, I knew I wanted to be a detective. I was absolutely convinced this was what I was going to do with my life. And of course I am, just not the sort of detective I'd read about in that book. And I use this as an example of how important it is to read about stuff that's way outside of anything you might be directly interested in, because you just never know when you're going to find something that is going to have a big impact on your life. And maybe you want to change fields. Maybe you don't like what you're doing and you read something and you say, wow, that sounds a lot better than what I'm doing. And I say, if you do that, don't hesitate to change. Don't hesitate to go and trace something that is really you feel passionate about. Because the people who are really happy in this life, they go to work on a Monday morning, but it's not really work. They love it so much that they just want to get totally engrossed in it. They're just passionate about it. And come Friday, they say, well, you know, do I have to go home? What am I going to do until Monday when I start work again? This is the kind of people that you can be, kind of person you can be if you're a scientist. Science is one of these careers like that. Just wonderful. I never consider I work. You know, my wife always tells me, oh, you're going to work today? I sort of say yes, but I know I'm not really going to work. I'm going to have fun for the day. So that is something I learned fairly early on. But then my father bought me one of these chemistry sets. And you buy a chemistry set today and it's got all sorts of very dull chemicals, and the experiments they suggest, maybe they change color, but that's about all. But I discovered that the chemistry set they were selling when I was 11, you could make explosives. You could make fireworks out of these chemicals. And I fell in love with chemistry. I've always loved chemistry as a result of this, as a result at age 11 being able to make fireworks. And I might say, I still have all my fingers. Now, this was the grammar school I went to, the City of Bath Boys' School. And they had a very good math teacher there, and I just increased my love for mathematics. I learned to play the violin and joined the school orchestra. And this turned out to be important, not at that time, but later on in life. Because one of the things about music, if you play an instrument, you have to focus on everything so much nothing else matters. It takes you away from whatever else you might have been thinking about. And I love that about it. I thought it was just great. I was also very close to the Mendip Hills, where there are caves. It's a limestone escarpment, terrific caves. And caving also I learned something important from. I used to go into the cave, and there was sort of a standard path, and the leader would take you down the standard path. I always wanted to go up into the little crevices on the side, and maybe you could see a chimney going up, and I would climb up and find. I wanted to go somewhere new. I wanted to find something that no one had seen before. And again, this has driven my entire research career. I also discovered jazz. Bath had a very famous jazz festival. That was fun. Now, when it came time to go to university, in England, you took A-level exams, and if you did well, then you could apply to any of the universities. And I failed physics. The first time I took it, I did not like physics. We had a terrible teacher, and I failed it. And part of this was also that I had gotten kind of bored at school. They were not teaching stuff very well. And I used to go down to the local YMCA, and I would play billiards and snooker during the day. And then I'd go back to school and sign out and pretend I'd been there all the time. And I thought I was going to be thrown out. They threatened they were going to throw me out of school. And I went and thought, well, you know, what else could I do? And playing snooker and billiards, this was what I did best. I was the West of England junior champion. And Joe Davis, who at the time was the world billiards champion, was looking for a sparring partner. So he made his money by going around and doing demonstration matches. And so I went to audition to become his sparring partner. But then the headmaster relented and said he wasn't going to throw me out of school, And I always felt I've really missed my chance for fame. I think I could have been the world snooker champion if I'd followed through properly on that. Now, when I went to Sheffield and did chemistry, it was very, very nice. It was really easy. We had to do physics, chemistry, mathematics. All of a sudden, we had a teacher who taught physics properly. I discovered that physics was really just all mathematics, so that was fine. I had no problems with that. And then we had an organic chemistry professor, W.D. Ollis, who was just a fantastic lecturer. He used to teach chemistry as though it was a series of puzzles and problems. And when he used to give exams, had Google been around and you could have taken your computer in, Google wouldn't have known how to answer these questions. You had to have really understood the chemistry that he was teaching. And I loved it. It was great. And so when I finished my bachelor's, I didn't get a first-class degree. I got an upper second. But I went to, Ollis said I could go be a student with him, so I worked in his lab. But what happened there was something rather interesting. This man, Kazu Kurosawa, this is a picture of him about 5 years ago when I met him in Japan, but at the time he was a postdoc in the lab. And I learned more chemistry from him than anybody. He was fantastic. He was such a good teacher. You know, he would say, well, I think you should do this, and I think you should do this, and he'd explain why you were doing it. And by the time you'd finished, you knew why you had done this experiment, and almost always it worked, and so this was good. And I just learned so much. He was fantastic. He's the best mentor I've ever had. He also taught me how to play Go, this Japanese game on the left, and shogi, Japanese chess, and we used to play by mail for a while until his English got so bad that he gave up on sending me mail. But the single most important thing that happened during all of that time after mentorship was I kind of got a little bit bored because I had all the data I needed for my Ph.D. in 1 year. And I used to go over to the library and read about stuff, and I read this book by John Kendrew called The Thread of Life. And it talked about the very origins of molecular biology, how it all got started. And I was just hooked. By the time I finished that book, I knew I had to be a molecular biologist. And I talked to people in the chemistry department, and with one exception, they all said there's no such subject. You know, it's nonsense. This is nonsense. Remember, this is back in 1968. But this one assistant professor said, you know, he'd heard about it and it sounded good. And so I applied as a postdoc to 6 different labs, all of whom were doing not deep molecular biology, but they were on the fringe of organic chemistry and molecular biology. They all turned me down except for one person. And the person who took me was Jack Strominger. And he was at Wisconsin, University of Wisconsin, Madison, Wisconsin at the time. And so I thought I was going to do a postdoc in Wisconsin. But then a month or 2 before I had to go, he called me up, or wrote to me, I think, and said, you know, could you just delay coming for 2 months because I'm moving to Harvard. And so here I am, quite by chance, going to Harvard instead of going to Wisconsin. And while Wisconsin is a good school, Harvard is a fabulous school. I really had a good time there. And Jack had just become professor. This has been his dream to be a professor at Harvard. And he had been working on bacterial cell wall biosynthesis, and there were a few projects to clean up, but his interest really was in cell biology in the immune system. And so he started afresh at Harvard, changed fields completely, started working in the immune system. And I was one of a few people left cleaning up the bacterial cell wall story. And so he wasn't really on top of me all the time because his interest was somewhere else. And so I had the opportunity to do things for myself. And I think this is something else that's really very important for students. If you find good students and they're working for you, let them do what they think is good. Don't try and tell them what to do all the time. The good people, they come up with great ideas, they do wonderful things. And in this case, the person that I picked the project up from had been trying to sequence this tRNA using what were the current techniques at the time, but they were not the most advanced techniques. The most advanced techniques were being developed by Fred Sanger, this man in the middle. He's one of the few people who have won 2 Nobel Prizes. He won the prize for sequencing proteins, and later for sequencing DNA, which he shared with Wally Gilbert. He could also have won a 3rd prize for sequencing RNA. He developed all of the methodology that was necessary to do this. So I went to his lab, learned how to do it. He was a very modest man. When I first met him, I went into the lab and there's this little guy there who's washing the dishware, right? And so I say, you know, hello, I'm Rich Roberts. I've come to work with Fred Sanger. And he looked at me and he said, hi, I'm Fred Sanger. And you would never have guessed. And he was very humble, but incredibly smart and full of good ideas. And it was just a joy to be with him, near him. And I've always felt, you know, if I had a hero and I wanted to be like someone in science, it would be Fred. He was a great guy. Now, something interesting happened after I'd been there for a few years. A lot of the people came to my lab to learn how to do sequencing because I was the first person in the Boston area to be using Fred's technique. And one of the assistant professors, Mark Potashny, came up to me in a seminar one day and he said, look, Jim Watson's going to come and talk to you because he has a job down at Cold Spring Harbor that he wants you to do. So, you know, I thought, fine, and so I waited, and then a couple of weeks later, Mark comes by and he says, well, Jim doesn't actually know who you are. Could you go and introduce yourself? So I go and introduce myself, go into his office. His opening words were, I want you to come and work at Cold Spring Harbor to sequence SV40 DNA. Right, this was the opening words before he, you know, whatever. I was in his office for 10 minutes, he was rambling on about something, and then I was just walking out and he said, oh, you probably should come down and visit Cold Spring Harbor and give a talk about what you've been doing, you know, but you've got the job, don't worry, you've got the job, don't worry about this. So that's how I got to Cold Spring Harbor. Now this is where things start to get interesting. So he wanted, as they just started, they had a big cancer center grant, and one of the things they wanted to do was sequence SV40. But there were 2 other groups already doing it. And I've never really liked competition. You know, there are 2 other groups doing it. Why would I want to start afresh and try to do it? They were going to do it. And so I thought, well, maybe there's something more interesting or different that I can do. And I listened to a lecture by Dan Nathans, the person in the top left. And he was talking about this enzyme, a restriction enzyme. At the time it was called endonuclease R. And it had been discovered by Ham Smith in the bottom left-hand side. And it turned out that if you took SV40 DNA and treated it with this restriction enzyme, it gave 7 or 8 nice fragments. And since the original discovery of HindR, there were a bunch of other enzymes, maybe 3 or 4 other enzymes that had been discovered that recognized different sequences. And so I thought, well, maybe if there are more of these, that this could be used as a way of sequencing DNA. And so I went to Cold Spring Harbor, I made the enzymes that were already known, and started looking in new strains. And everywhere we looked, there were these restriction enzymes, and they all recognized different sequences. And so I thought, here's my opportunity. I can outdo Fred Sanger. I'll come up with a method of sequencing DNA that's based on restriction enzymes. Well, needless to say, it didn't work out. But on the other hand, the restriction enzymes turned out to be incredibly important. When we were first discovering, I think we discovered maybe 25 of the first 30. And we had them all in my lab, had them in test tubes. People used to come to meetings at Cold Spring Harbor. They'd have a tube of DNA in their pocket. And they come down to my lab and they say, oh, do you have something that will cut this? And we got it all set up. They could just do the experiment themselves, set up the digests, run the gel, and then they would know. And usually they found something that they wanted, and then we'd give them a sample of the enzyme. They'd take it back with them, or we'd give them the strain if they wanted to make it themselves. And it became apparent that this was something that an awful lot of people wanted. And in fact, in retrospect, thinking about what people were using these for, this was the origin of recombinant DNA. People were making these little fragments with restriction enzymes and then joining them together. And this was how SV40 became a vector for various things. All the plasmid DNAs were discovered, and they were all using restriction enzymes to cut everything together. And it was obvious there was a business here, that you could make money doing this. And so I go and talk to Jim Watson, say to Jim, why don't we start a company, sell these restriction enzymes, and we'll use the profits to support the research at Cold Spring Harbor. To which Jim said, oh, it's very dirty to be associated with industry, and plus you can't make money that way. Just dismissed it. He wasn't even interested. But I knew that there was something, and the point was the scientific community needed these enzymes. They weren't going to make them themselves. There were hundreds of these. And so I started asking around, and I was pointed to a company called Miles Laboratory, who were kind of a sigma at the time, and they were sending reagents, selling reagents. And I called them up, and they said, yeah, we're thinking about selling this endonuclease R that Ham Smith had discovered. And I said, but there are loads more. Wouldn't you rather sell some of them? And they said, well, it's not actually us. It's this guy, Don Cohn. He's actually a company called Biolabs at the time. And they said, give me his phone number, and said, why don't you talk to him? So I called him up, and he was very interested. And he said, why don't you come up, talk to me? And I really gave him all the strains that he wanted to make the enzymes. We gave him the protocols. We did the quality testing. He would send the enzymes down to Cold Spring Harbor, and we would check for quality. And the company got started in a major way. He offered me a partnership in this company, but I didn't really want to be a partner. I still have my Nobel Prize winning work to do at Cold Spring Harbor. But I've never really been interested in money anyway, other than if it can be used for good purposes, right? Good humanitarian causes or supporting science. And Don also wanted to use the profits to support science, to support basic research in biolabs. And so anyway, so the company got started. Within a year, a little over a year, they had made $200,000. This was back in 1975, 76. And they gave me 10% of all the sales, okay? And Don had originally thought he was going to price these enzymes at $40 for a little tube, and he upped it to 44, 4 for me and 40 for him. And after 2 years, this just got to be ridiculous. I mean, I was getting so much money, it was crazy because I didn't feel I was really doing much. And so we renegotiated, and I said, well, let's do it 1%. And then after that, it became, when that got to be ridiculous, we just settled for a discrete sum every year. And I mainly used that money to support things at Cold Spring Harbor. But on my floor, not more generally, Jim had been so nasty about it. Anyway, so we proceeded this way, and I worked at Cold Spring Harbor until 92, and at that point I was fed up with Cold Spring Harbor, and I was assistant director, and there was nowhere to go because Jim didn't want to hand anything over, and he and I were fighting all the time. So I thought I'd start a company to sequence DNA. This was the time when people were thinking about the Human Genome Project. And so I, at that point, looked around, found some people who were interested in putting some money into a sequencing company, and then Don said, well, if you're going to go to industry, why don't you come to us? And so he made me a very good offer, and that's what I did. I moved up there in 92, and then a year later won the Nobel Prize, and I've been there ever since, and regretted not having gone up earlier, because the last 6 years at Cold Spring Harbor with Jim Watson were pretty awful. It would have been much better to go and do something more fun and more interesting. Now, one of the things that happened at Cold Spring Harbor was that Jim and I would quarrel all the time. I mean, he got really pissed off when I didn't sequence SV40 using all the RNA that he claimed he'd hired me for. And so he used to come, I remember one time, very early after I'd gotten there, I was in the kitchen where the ladies were washing the dishware and making media and so on. And he comes and starts shouting at me and telling me how useless I am. And this is sort of typical of Jim's style. I mean, he likes to bully people into doing things that he wants them to do. So he and I fell out all the time, and Jim was constantly threatening to fire me. But this gentleman, Norton Zinder, who is probably Jim Watson's only really good friend ever, but he liked me. And he used to, every time Jim threatened to fire me, I'd call Norton, Norton would talk to Jim and convince him, keep me on, keep me on. So I owe a great deal of my career to Norton. Now, one of the things that happened as we were discovering the restriction enzymes, we wanted to find things to do other than, just figure them. And so this postdoc, Richard Gelinas, came to my lab. He'd been at Harvard. I'd met him as a student at Harvard. And he came down, and all of the postdocs who came to my lab, the first thing they did was find their own restriction enzyme, but then they all had a project that might be more interesting going on. And so he was very interested in asking the question, do eukaryotic promoters, the regions of the DNA that tell the RNA polymerase where to start making RNA, are they the same in higher organisms as they are in bacteria? Because Mark Potashny and Wally Gilbert had figured out what they look like in bacteria, but no one had figured out for a larger thing. So we started working on adenovirus in 1975. There was quite a lot of transcription work had been done, partly as a result of mapping SV40 and then mapping where the RNAs were onto it. And actually Phil Sharp, who shared the Nobel Prize with me, and this was our only paper that we had together because he moved soon after and went up to MIT, and our work was completely independent after that. So we started working on the early RNAs, E1A and E4, because we thought if we can figure out where the RNA is starting, we can sequence the DNA coming in from the end, and eventually we're going to hit the sequence where the RNA is starting, and then we'll know what is the signal that tells the RNA to start here. Well, we worked for quite a long time on that, but we couldn't get enough RNA in order to really do the experiments. And the experiment we were trying to do was a very straightforward one. In eukaryotes, the messenger RNA has this poly-A at the 3' end. It's put on separately by an enzyme after the RNA is made, and so that's not coded on the DNA at the end of the RNA. And at the 5' end, at the time, we knew there were triphosphates, so all RNAs we knew about started with triphosphates. Ribonuclease T1 is an interesting enzyme. I'd learned all about this when I was sequencing RNA with Fred Sanger. And it cleaves after every G residue. And so if you take a full-length messenger RNA and cleave it with ribonuclease T1, you get 3 kinds of oligonucleotides. You get the one that's got the 5' phosphate. This is the 5' end. You've got the poly-A, which we knew was a 3' end. And all the others are just something G, something G, and so on. And so we tried to find a way that we could separate these nicely, and in particular to capture the 5' end. But we really didn't get very far trying to do that because we didn't have a good way of getting these 5' ends. But then we also switched to late RNA because the early RNA didn't work. We couldn't get enough RNA. But late in infection, almost all of the transcription that takes place when adenovirus infects a human cell is late infection, and they're all shown here, L1, L2, L3, L4, L5. We knew there were a dozen or 15 different transcripts. And so we also had another piece of luck, and that was that a group discovered that there was something funny about the 5' end of eukaryotic messenger RNAs. They had this funny cap structure. So the triphosphate, instead of just being a triphosphate, actually had another oligonucleotide, a modified G residue, that hooked onto it, but in reverse orientation. And so it had 2 hydroxyl groups sticking down here. So it really looked like the 3' end of RNA rather than the 5' end. And now you can get rid of the poly-A really easily by hybridizing it to oligo-DT and running it through a column. It all disappears. And so now we had a way to find how can we get the 5' cap structure. And so I came up with what I thought at the time, and I still think, was a very clever experiment, using a boron derivative in order to catch this oligo. And the idea was that if we could do this, then we could do a fingerprint of all of these 5' ends, and they would tell us where all of the late RNAs were starting. And so we could actually look and see what was the distribution of all the cap structures. And so Richard, and here's the experiment. So we look for all the ones at the bottom, but using the advantage of this cap. And so Richard did the experiment and got this result. So we were expecting 12, 15 spots in a 2-dimensional array. but there was really only one. It looked, to start off with, as though it was two, but they were just related in that one was a cyclic version of the other. And if you let it go a little longer, there was just one spot. And that said, all 15 RNAs had exactly the same 5' end, which didn't make sense, right? They're scattered all the way along the adenovirus genome. How can they all have the same 5' end? And so, I told Richard that when he first produced this result, I said, maybe you better go back and do it again and see if you can do it a little more carefully this time so that we capture everything, which he did and got the same result. So I said, Richard, why don't you sit down? I'll do the experiment. I'll show you how to do it properly. And I got the same result. So here we are. We have a result, totally unexpected. This was a failure, I was expecting 15 spots and there's one. Our experiment failed. And it's always worth remembering that when things fail, it's either because you screwed up when you were doing the experiment or nature is trying to tell you something. And in this case, nature was trying to tell us something, but we didn't know what it was. It took quite a while for us to figure out what it was. So we published a paper describing this. It was an 11-long oligo, one predominant undeconucleotide at the end. But really, what did this mean? How is it possible that you could just get this? And so there were various theories coming along. One was that it was just a repeated sequence all the way through, so that every time this sequence was there, it said start making message. But then we did a series of other experiments, and we had some people who came along. And it didn't matter where you hybridized the messenger RNA to the DNA, there was the same cap structure on all of them. Pure RNA for several of the messages were found, and they had the same cap structure on them. When you hybridize the messenger RNA, though, to its coding region, and just treat very gently with a ribonuclease to try to get rid of all the stuff that hadn't hybridized, you lost the cap structure. It disappeared. So if it had been hybridizing to the DNA, it should have stayed there. There was no reason for it to disappear. Saeeda Zain was a Pakistani scientist who was a postdoc in my lab at the time, very talented young lady, and she showed for SV40 DNA, where there was adeno-SV40 hybrids, that again, the cap was not present on the adenovirus part of it, and it was really weird, very strange results. And also all the early RNAs we later discovered, not at the time, they all have a different cap structure on them. So what's going on? Well, one way to think about it is if you look at this schematic of what a structure looks like. So here's the DNA at the bottom. You take the RNA, you hybridize it to the DNA, so all the bit that's coded there forms a nice hybrid. At the 3' end, the poly A is going to be sticking out. And at the 5' end, something is sticking out. Something has a cap structure on it. So again, how do you get there? And we had ideas. The one I favored was one that Eric Davidson had proposed some time ago, was that there was a primer. And the primer was actually getting the RNA, and it knew where to hybridize, and it was getting the RNA made. But there's a nice experiment that we came up with, and it's called an R-loop experiment. Turns out if you take messenger RNA and double-stranded DNA and heat up the double-stranded DNA so that it melts slightly, you can form a hybrid between the RNA and the coding strand of the DNA. And because RNA-DNA hybrids are more stable than DNA-DNA hybrids, this was a stable structure. And so you can make these, and then you can figure out what you're going to do with it. Well, when you see it, you're going to see the poly A off to one end, and someone had already shown that there were ways to detect that. And so the question was, how are we going to detect the thing at the 5' end? Well, the obvious way is to take a piece of DNA that codes for it, and then add it, and then look and see. And this led to the idea for this rather nice R-loop experiment. DNA probe to look at the 5' end. So I came up with this idea on a Saturday morning. Actually, Richard and I, we'd gotten into the habit, we'd had a post-mortem every Saturday morning about what had gone wrong and why things hadn't worked and how we were going to really do the key experiment the next week. And Richard was busy explaining something, and all of a sudden this idea came to me. And I said, Richard, sit down, why don't we do it this way? And he looked at it and said, yeah, that looks okay. But it was an electron microscopy experiment, and neither he nor I were electron microscopists. But fortunately, just down the hall, there were two excellent electron microscopists, and they were in on this Saturday morning, and we went and talked to them and said, we have this idea for the experiment, and I knew what I wanted to use for the DNA probe because I had an idea, a guess as to where it was being coded. And I said, can you do the experiment for us? And they looked and they said, well, it's not quite been done before, but yes, we could do this. And so Richard made the reagents on the Sunday and the Monday, and we took the stuff down to them to the Tom and Louise on the Tuesday. And the very first molecule they looked at in the EM looked exactly like this molecule that I'd drawn out on the blackboard. Well, not quite the same. If you look in the middle, this is the picture that I'd drawn in the previous slide. At the bottom was what they actually saw. So instead of the probe hybridizing at just one position on the RNA, it was hybridizing at two positions. And so this said that this structure at the 5' end of message actually had two separate pieces associated with it. And when they did more, when we used other probes, there were actually three. There were three little segments, and they were tied to all of the late messenger RNAs, no matter where they started. And so this was basically the discovery of RNA splicing. There was a lot of work went on after that. Tom and Louise did a great deal of experimentation, doing electron microscopy, which we couldn't do, and so we mainly did biochemical kinds of experiments or molecular biology. But this was the paper that described it, and you'll see it's got the term amazing in the title. And we had 3 referees, and each one of these referees wrote back and were saying, these are nice experiments, and they had some nitty-gritty things, but they said, you can't use amazing in the title of a paper. This is not a scientific term, it's not magic. And so Ben Lewin, who ran Cell at the time, was the editor, who I knew a little bit. And so I called him up and started talking to him about all the other things that the reviewers had said. And so halfway through the conversation, I said, what did you think when you first heard this? And he said, oh, it was amazing. I said, I rest my case. And so there it is. So apparently this was the first time, or so I'm told, that amazing was used in the title of a paper. So after the discovery from 78 going on, we sequenced a bunch of spliced junctions where these splicing events were taking place. But it soon became apparent that we needed to sequence the whole DNA because there was so much splicing going on. And so we did that. And in order to do that, we needed programs that could put the little pieces of sequence together so that we could eventually assemble it. They didn't exist at the time, so we wrote the programs to do that. I learned Fortran so that I could program some of this. And we got another guy involved in it. And so that worked out very nice. And then we started developing bioinformatics methods to actually look at the sequence and figure out what was in there. And so that got me hooked on bioinformatics. But the combination of programming and what was going on and my love of mathematics, it all came together. It was great. And here we are also being a detective. So, I just had the best of everything. So this was the prize. And I've got to say, the Swedes really know how to throw a party. You go there for about 10 days, and every day there's a lunch and a dinner that if that was the only thing that happened during the year, it would have been the highlight of the year. So much champagne is circulating, it's very difficult to stay sober for any significant length of time, but it's wonderful. I recommend it to everybody, it really is good. Now, the other thing you have to do is you've got to give a lecture, a Nobel lecture. And most people get up and they talk about all the wonderful experiments they've done and discoveries they've made following the original discovery. But I'd stopped working on it many years previously. But luck came my way again. Just before I had to go and give my Nobel lecture, I was working with this crystallographer, Xiaodong Cheng, and we've been trying to get the structure for a DNA methylase. And this was a picture that came out when it was binding to DNA. And what it showed out, the yellow are the DNA bases, and the base that is going to be methylated is flipped 180 degrees right out of the helix into an active site in the enzyme. This was the first discovery of base flipping, as we called it, and it was obvious that you could do many reactions. Anytime you wanted to do chemistry on a DNA base, you had to get at it, and this was the easy way to get at it. And we now know that almost everything that does chemistry on a DNA base uses this mechanism to get it out. And so this was my Nobel lecture, and there is no better place to talk about a new discovery than to the Nobel Foundation. So let me go and tell you a little bit about New England Biolabs. They were founded in 1975. When I joined them, there was Don Combe, Marilyn Combe, and one technician, and they were operating in a basement in Beverly underneath a hairdressing salon, and they got used equipment from Harvard. Don had put most of his savings into setting this little lab up. The rent was, I think, $180 a month or so. It was next to nothing. And he was also doing some pregnancy testing to earn money and going lobster fishing. But he switched completely. We got the lab going, and we were in that lab, and then upstairs we got some space for probably about 4 or 5 years, and then we moved to another building, and now we're in another building altogether. And so since 1975, when New England Biolabs was first incorporated as New England Biolabs, we've gone from 4 employees to about 630 worldwide now. We have 330 in Ipswich in the main campus, and of those, slightly over 100 people are doing research, most of it basic research, and all paid for by the profits from the company. And it's an amazing place to work. People who get a job there and work there, they never want to leave. We had one case of a lady who worked there, and her husband got a fantastic job over in California. And she told him, I'm not going, You go by yourself. I like what I'm doing. So he eventually decided he would stay where he was. So that worked out pretty well. Now, I moved up there in '92. Myself and others cloned restriction enzymes for commercial production. I'd been involved in that actually for a number of years previously. But we started to really try to explore the limit of bioinformatics. I have a couple of programmers who work for me, and they were busy doing a lot of work there. And then we did a lot of genome sequencing initially, just sort of to develop better software to put sequences together, but then also to try to find new restriction enzyme genes. And that worked out pretty well. Now, in 2010, Pacific Biosciences came up with a super way of sequencing DNA. They could get very long pieces, they were very accurate, and it was easy to put them together and assemble genomes. And, we're at a point now where in basically 1 day's work, using the Pacific Biosciences method, you can close a bacterial genome, sort of 2, 3, 4, 5 megabases, they just close. The thing I'm interested in is it also tells you what sequences are methylated. And for me, this is wonderful because methylation is how cells protect themselves against restriction enzymes. And so this gave you the sequence of the methylase, and pretty much every case we know of, the methylase sequence and the restriction enzyme sequence exactly match, as you might expect, because the methylase is protecting the host DNA against the restriction enzyme. And so that's been incredibly successful. We've seen that one. So what have we learned so far? We've learned that there are many solitary methylases that are not associated with restriction, that are present in bacteria. 1 or 2 of them, we know what they do. Most of them we don't. Most of them we have no idea. A lot more variability in recognition sequences than we were anticipating. The type 2 methylase specificity usually, I put sometimes, but I should say usually, matches the sequence of the DNA restriction enzyme. And then we've got all these many non-specific and other methylases that are there. And I can just tell you about one very recent discovery. We found that in Clostridium difficile, C. difficile is something that if you go to hospital, it's very easy to get infected with it. These things are everywhere. And C. difficile, first of all, it's got a lot of antibiotic resistance, but then if it can't deal with it that way, it forms spores. So it just forms a little body, stops growing, sits there, waits for the antibiotic to go away, and then starts growing again. And so the only reasonable treatment that's come up for that is something called a fecal transplant, in which you basically take shit, isolate the bacteria from it from a healthy person, and then give it to the patient. And that actually is remarkably successful, but it's kind of a messy way of doing it. And a lot of people don't want to do it. They don't like the idea of, taking feces and putting bacteria from them in their mouths. So it really would be good if there was a better way of doing it. And so we discovered that there is a methylase that recognizes C followed by 5 A residues, methylates the last A residue, and if you knock that out, C. difficile can't form spores. So this methylase is driving C. difficile in order to make spores. And there are a couple of companies now down in the south who've found, they found small molecules that they think is going to be good enough to inhibit just this methylase, and there are 2 clinical trials underway. And the reason this is important is C. diff is absolutely awful. It doesn't kill you, but it gives you uncontrollable diarrhea. And so people get sent home, patients are sent home. They never want to leave their house because they don't know when they're going to suddenly have an attack. You don't want to be on a bus or in a taxi or walking and suddenly discover that you have diarrhea. So people just stay home. So this could be quite important. And I think it's just an example of one of many things that this could be good for. Let's skip that. Tell you a little bit about myself. I love puzzles and games. I spent my Nobel Prize money put in a professional croquet lawn in front of my house. I've been stud muffin of science on the left and various other things. I just love puzzles, games, and this all dates back to when I was 8 years old and this headmaster came and started getting me hooked on puzzles. I also got knighted by what is now King Charles. At the time he was Prince Charles. I was glad that his arm was pretty steady because, when you've got a sword right next to your neck, you want to be a little bit thing. Several buildings got named after me. I got knighted. It was the 2008 birthday list. But one of the things that happened that I just would take a few minutes to tell you about is that before I won the Nobel Prize, I had lots of good ideas and, I could run the world and suggesting to people what they should and shouldn't do. And people, sometimes would listen politely or they just walk away, but they almost never did anything about it. And I discovered that after I won the Nobel Prize, people started listening to what I said. All of a sudden, I'd gotten some glamour or I don't know what it was, I'd gotten something anyway. And so I thought, I talked to other Nobel laureates and they sort of said the same thing. And I thought, well, maybe we could organize the Nobel laureates to start doing some good. And there are really 2 issues that we all pretty much agree on. One is we would like to use our notoriety, if you like, to do some good humanitarian work. And the other is to dispel myths about science. There's so much stuff, misinformation about science. And now with social media, it's just everywhere. And people get scared about things. The GMO campaign is a good example. But I really found out how it was possible to really have a big effect as a result of this incident in the Children's Hospital in Benghazi, Libya. So HIV was spread to about 400 kids there, and there were 5 Bulgarian nurses and 1 Palestinian student who were working there, and Gaddafi decided they were the criminals. They were doing this deliberately, put them in jail, they tortured them, they got some confessions out of 2 or 3, but almost everybody who knew how the hospital system and nursing and so on, medical practices in Libya were absolutely convinced that this was not the explanation. They weren't deliberately spreading it. It was just that they reuse needles. They reuse hypodermic needles because they didn't have enough of them. And so there was a lot of work done actually examining the strain that came and looking at what was going on. And, you know, it had never occurred to me that people would allow Gaddafi to take it as far as he did, but then all of a sudden a Nature paper, a Nature correspondence piece came saying they were on death row and they were going to be executed imminently. And I got outraged. So I got a load of Nobel laureates, I think we got about 120 altogether, who signed a letter that we sent to Gaddafi. In the meantime, I'd actually engaged the Foreign Office in London, and they were doing nothing when I first contacted them, but then they started to do something. And this letter I took down to the Libyan ambassador to the United Nations, because the US didn't have any diplomatic relations with them at all. And I presented this letter to him and told him what had gone on, and I asked if he would give it to Gaddafi. And so he was very nice, and 2 weeks later I got a telephone call one day from Gaddafi's son, Saif al-Islam, and he called me and he said, could you come to Tripoli and talk about this? So get on a plane, go to Tripoli, meet with him, and at the end of the conversation, he said, we're going to let them go. We know they're not guilty. And so they were let go, and we got them out. And I thought, you know, if a few Nobel laureates can do this, what else can we do? Surely there are many other things we can do. And so I've been involved in a lot of campaigns now about that. The one I just mentioned briefly is the GMO campaign. Here in India, you have a lady called Vandana Shiva, who is just going about lying right, left, and center about the dangers of GMOs. She charges $50,000 for a lecture, which I'm sure is why she's doing it. She's very popular. A lot of people follow her. But she and many others, Greenpeace and so on, they've been spreading lies about GMOs. GMOs are safe. You know, the science, for 30 years now we've been growing these. People have been eating them. No problems. Not a single problem with any of them. You can't say the same about plants that have been derived by traditional breeding. Traditional breeding is really a dirty way of mixing genomes, right? Because you get all half genes from one place, half from the other. It's really only one or two that you're usually trying to select for. The GMO method allows you to select for them. And I like this cartoon, you know, the sort of anti-GMO people telling everybody, oh, these are really dangerous. Do you know what these things can do for you? They'll kill you. But the little boy in Africa says, I know exactly what they do. They keep me alive. And I think what's happened is that we've reached a situation in many of the developed countries where we say things for political reasons, for money-making reasons or whatever, and we think it doesn't have any effect elsewhere. And I say if the Europeans don't want them, then don't eat them. But please don't tell the developing world that they're dangerous. And, you know, India has taken a long time to come around to developing GMOs and allowing them to be grown. BT cotton was the first, and the cotton farmers loved them because all of a sudden they're making a lot more money. You don't need as many pesticides. And as you were saying last week, mustard was approved. And we hope that more like that will come. So I think there's a lot of hope there. Now, I just want to mention the importance of luck. So I learned this lesson, I think I was 16, maybe 17. I was playing in a tournament, a snooker tournament, played my shot, and the ball that I was trying to get in the pocket didn't go in the pocket, but another ball did, one that I wasn't aiming for. And I'd make the next shot, and I muffed it. It just didn't work. And afterwards, an old gentleman, must have been 80, came up to me and he said, you know, something you have to learn is that when you have a piece of luck, you've got to make the most of it. You've got to try twice as hard on the next shot to make sure you don't waste the luck that came your way. And this is really good advice. I've taken this to heart. I look at it all the time. I think about it all the time. And I think you might, at some point, you know, one evening you've got a little time, look back and see how luck may have influenced your lives, and did you take advantage, or did you feel guilty and not really take advantage? And a lot of people just feel guilty. And Everybody has luck, no reason to feel guilty, make the most of it. Kazu Kurosawa, the postdoc at Sheffield, who really taught me all about chemistry. I had no idea he was going to be there, didn't know he was going to be a great mentor, just showed up purely by chance. Going to Harvard instead of Wisconsin, another lucky break. Looking for eukaryotic promoters in adenovirus. We didn't know what we were going to find. And what did we discover? We discovered one of the really important principles of molecular biology, for which eventually I got a Nobel Prize. But that was luck, total luck. We weren't looking for it. It just came our way. Perhaps the best is taking an early plane. You probably all know about the plane that went into the World Trade Tower on 9/11. That plane I was booked on until 2 weeks before it took off. I was going to a meeting in California, and the meeting got moved forward by 1 day. It was actually a meeting that NASA was organizing. I was on their astrobiology committee. And so I took the plane the day before. I wake up in California. I see the plane I would have been on going into the World Trade Tower. That is luck. That is luck. And I've really blessed every day that I've been alive since then. It was just fantastic. And so I owe that to NASA. So that's pretty good. And studying bacterial RM systems, again, that's turned out to be really pretty good. So I just want to acknowledge Richard Gelinas, Louise Chow, Tom Broker. These were the people who helped get the experiments done to prove splicing. Saeed Zain and Dan Klessig, these were people who did really made important discoveries, but it was data that they produced that we could use to really argue to ourselves that what we were looking at was real. Huge number of lab members, colleagues, collaborators, friends over the 50 years that I've been in science. You just can't beat having good collaborators and having good people in your lab. And finally, Karen Otto is my assistant, my secretary. She worked for me for 37 years. She just retired at the end of September. I don't know what I'm going to do without her. So thank you all. Yeah, sure. It's just that I went over a little over what I was asked to do. Anyway, go ahead.
Okay, so before we start with the session of questions and answers, I just want to make a few requests. Please wait for somebody with a mic, or for the mic to reach you and speak into the mic clearly so that everybody can hear your question. So I think we can start. Oh yeah, another. So given that we actually have organized this talk mainly to encourage young students, we would request that the students be given a chance to ask questions, as many as they can. Thank you. Anybody who wants to ask something can raise their hand. And if there are students in the other seminar hall who are watching the live streaming, they can join us here if they have questions to ask.
I would say one other thing. If you have questions and you just want to email them to me, it's roberts@neb.com, and I usually answer everything that comes my way. Yeah, go ahead.
Yeah, hello sir, this is Ankita here, and it was really an inspiring talk. So I would like to know about the splicing mechanism, and first of all, nowadays the non-canonical splice sites are also coming up in the case of cancers. So we in our lab are trying to generate some mini genes with respect to know what would be the impact of these non-canonical splice sites in the cancer, and also trying to identify some of the novel transcripts which may involve in the cancer. So
okay, so I'm not the right person to answer that question because I've not worked on splicing since 1979, 1980. And there's been so much knowledge generated, and quite a lot of is known about how splicing takes place, what's important. but I'm just not an expert in that area. So, but there are plenty of people who are. I have a postdoc, Adrian Craner, at Cold Spring Harbor Lab. If you asked him, he might be able to tell you. He was the first person to actually make a medical discovery breakthrough as a result of splicing for spinal muscular atrophy. He developed a mitigating oligonucleotide because it's a defect in splicing, in alternative splicing that takes place. But he would be much better to answer a question like that. His email address is craner@cshl.org. He doesn't know that I'm giving out his email though. Question over there.
So it's very great pleasure to hear you and to know whole the journey of your life. So thank you so much for that first. And just now you said about astrobiology in NASA. So I just wanted to know how the biologist can explore their knowledge in astrophysics through NASA. So if you can guide a little about that.
Okay, so NASA got very interested in the idea that there might be living organisms elsewhere in the universe. And the obvious place first to go looking was Mars. But because of the atmosphere on Mars, it would be unlikely there would be anything on the surface, and you would have to dig under. And then there are various moons of Jupiter and Saturn that also look as though they might have life on. And so NASA were interested in getting some advice about how they would detect life. If there was life, what would it look like? And I actually have another talk I give about why I love bacteria, and I always end up with a picture of Mars. And I'm always saying, I hope we discover that there is some life on Mars and that it's older than life on Earth, because then we'll all be Martians.
I think it's...
So far there's no evidence for anything, but NASA are looking.
So you talked about all the times luck came your way and you actually did something about it. Can you tell me about the times that you regret that luck came your way but you did not act?
Yeah, well, I tell you, in this snooker tournament, I regretted that I didn't act. But, there've been a number of times when something lucky happens and you just don't recognize it at the time because you're not looking for it, or maybe you're in a situation where you don't feel you should take advantage of it. And certainly before I was 17, I'm sure lots of luck came my way and I blew it completely. But I really took to heart, this gentleman, he was very persuasive about how important, and it is important. And I think it's worthwhile, everybody, just taking time, looking back and saying, well, why did I go this way instead of this way? And, was there a good reason or was it just luck? Because everybody has luck. I organized a series of symposia a few years ago, in which I got Nobel laureates to talk about a subject that I call serendipity in science. And there's so many of them. You talk to them and you say, well, were you looking for this? And they say, no, we were doing something totally different. It didn't work, and we went to see why it didn't work. And so it's always good to do post-mortem on failure, because very often nature's trying to tell you something. Lots of discoveries to be made, but I got to say, if you have to work in a field, biology is the best. There's so little we know about biology as compared to physics or chemistry or anything else. Way many discoveries just waiting to be made. Right? You heard earlier, 2% of the human genome actually codes for proteins. So what's the other 98% doing? We don't know. There's one here.
Sir, my question is, a little long, so please bear with me. Okay, a couple of years ago I watched this movie called Logan. it is a Marvel movie comic book characters which have their power because they have a mutation in their genome which gives them superpowers. In this movie, 10-20 years into the future, no more mutants, people with superpower can be born because a scientist used GMO crops to suppress this mutation. I mean, no more mutants could be born in this world with the help of a scientist who used GMO crops. So the applying GMO crops for food is like the first step. So what comes after that?
Well, I can tell you, Hollywood is very good at making up scary scenarios.
Well, sir, just in
If you want
case,
to see,
I'm not scared. I will
go
like
and see
GMO.
one of these horror movies, right? No, the whole point is that, I mean, just to give a simple example, almost all of the crops that are eaten and grown in the developing world, we don't eat, we don't grow them, we don't do anything with them in the West. You know, we have about 6 crops that provide almost all our food. And so the ag tech companies have done a great job in improving yields, improving everything they could. But nothing has been done for cassava, for instance, or very little for many of the crops that people eat. We can make sure that they grow better, that they become drought resistant, that they produce nutrients that they otherwise might not. I don't know if you've heard of golden rice.
I use that.
So golden rice is now finally approved in the Philippines. Okay? There's something like between 1 and 2 million kids who go blind and many die every year because they don't get enough vitamin A when they're growing up. And with golden rice, you solve the problem. So there are many examples like this. Why would you not do this? Why would the Europeans want to stop this? Greenpeace put out this booklet called Golden Illusion. And they said, well, it's taken so long to develop, it can't possibly work. They forgot to mention that they had done everything possible to stop its development. Everything. They've been a terrible organization on this issue.
Sir, can we use GMO for disease suppression?
You mean in humans, or in what sense?
Like to make humans,
Well, you could. Why would you want to do that, though?
no, like approving
eventually
GMO
we will
crops.
be able.
Eventually, in the future, we will have to get to the next step. I mean, we have better crops.
Yeah,
What's
but
next?
the way to think about it is that there are many inborn genetic diseases, right? Mother has a defective gene, father has a defective gene, and the offspring have it. It will be possible at some point in the future to correct that. We don't know how to do it today, though, because we don't have the accuracy that is needed to target just this one gene and know that nothing else is going to change. Chinese scientist who tried to do it because he thought he was going to be the first to get a Nobel Prize spent time in jail for trying to do it. So if you want to do it, then get your jumpsuit ready. No, I think it's just too early. We will be able to do a lot of things in the future, but we can't at the moment. And we shouldn't. We shouldn't at the moment.
Hello, sir.
Yep.
It was nice to go through your journey through the till now. So you just now said like GMO crops, right?
Yep.
So there is pros and cons to that also. So you talked about the BT cotton. So here in Indian state of this Maharashtra, so farmers are growing BT cottons from long time now, and now there is certain cases where it has been shown that there are certain insects like bollworm which now become resistant to the insecticide, pesticides, and that is now going to disturb the ecological balance as well as have environmental effect, right? So in addition to introducing the GMO crops to farmers, so we all know that there is certain steps and taken to prevent it from like doing such harm. But what your take on it? Like, is it that all this, so in this case that BT cotton has created this problem? So is it so that it is like a big environmental problem or the ecological balance is going to disturb because of this? What's your take on it?
Okay, so BT cotton has been incredibly successful. Pesticide use has gone down almost 1,000-fold as a result of it. But of course, eventually, insects will become resistant to this particular BT toxin. You have to realize Bacillus thuringiensis, various different strains produce thousands of different toxins, right? So we take one that the insects are not resistant to, and we put that into the cotton, and we develop new varieties. And plant breeders do this all the time. This is how you deal with the fact that things become resistant. It's exactly the same with human health. We need new antibiotics because bacteria become resistant to the antibiotics that we're using. But I don't think this is really a problem. It's something that we know how to deal with. We can deal with it. And so far, BT has been an amazing gene. In South Africa, in the southern part of the US, BT corn has been incredibly successful. And now a number of African countries are also trying to grow it, despite the fact that the activists from Europe have been going around trying to scare everybody. So it's a good thing, but these things happen. Nothing is perfect forever. Things change, but we know how to deal with it now. And the nice thing is the GM method is fast. Traditional breeding is slow. So let us say you wanted to make a new variety of cotton that was resistant to these bugs that were now trying to eat it. Typically takes maybe 20 years to develop a new variety. With GM methods, you can typically do it in 2 or 3 years. So the time difference is immense. Drought-tolerant crops are going to be incredibly important. Water-resistant crops, things that are not killed when the fields are flooded, these are going to be important. And these are things that we can do. But we've got to get the governments to stop trying to stop it and stop the activists from convincing people that they're dangerous. There's just no evidence for it. Why would you go around trying to kill off the developing world by feeding them misinformation? It's not right. I think it's criminal activity. It's bad.
Thank you.
So,
Okay.
so I have a question. Excuse me, sir. Excuse me.
Sorry.
It's an inspirational talk you gave, especially the Watson part I liked the most. Yeah. So regarding the GMOs, I have a question. These have done wonders, like rice nowadays growing in saline stress condition in the East Asia. But the thing is that, don't you think that genetically modified organisms, the genes that we modify in them, they might slip into the natural pool, the wild pool, and create undesirable effects as well? For example, one instance I would like to suggest. In United States, there was an attempt to control the mosquito population. So what they did was, they used that male population, the male mosquitoes were sterilized. And this was also done by the Chinese as well. They tried to eradicate, and they used that. It worked for a few years' time. The mosquito population was almost gone in that particular island. But later on, the growth was tremendous, and the nature took its course back again. So don't you think that these genes can slip into undesirable plants, weeds, invasive species, and create something difficult ecological balance?
Okay. But you realize that everything that is living on this earth is genetically modified. Everything. You are genetically modified. Okay? The bacteria living with you become genetically modified because they're exchanging plasmids all the time. So there's nothing special about what we do other than that we're doing it in a precise manner where we know what we're doing. And when resistant comes, then we'll find alternative methods. I've not heard that the mosquitoes were now coming back, because every experiment I've seen in which they've been trying to get rid of mosquitoes has worked incredibly well, and the people have been very pleased about it. They've been doing experiments in Florida, for instance. Okay. So, do you know what is the reference to that? So can you send it to me, roberts@neb.com? If you
definitely, sir.
Pardon?
I will send to you, sir. I have looked into it. I will send to you the reference. And the instance also, sir. And so another question regarding GMO, just want to clarify. So there is a catfish species called Clarius batracius. Now we Indians are used to have native catfish which we people eat here. Now recently because of commercial purpose and because I don't know about the experimental part, invasive species called African catfish was imported into the country because it gives higher yield. But what happened was it destroyed the native ecosystem, and I don't know, people used to buzz around, it might be a non-scientific part, and it resulted in the native species in being danger, and it eats up. So this kind of ecological disbalance which GMO might create, I'm just telling this.
Hey, I could scare the living daylights out of you with stories that might be possible.
Yeah.
But I can also tell you how to avoid them too.
Yeah.
And so, for instance, there are these GM salmon that have been developed, right, that grow twice as fast as regular salmon. And they've just been approved in the US after 20 years. And they're going to grow them under enclosed circumstances.
Okay.
And most things that get modified, when they get out into the wild, they're destroyed almost immediately. It is not the case that things that are modified always do better. Some do better, some don't, but a lot of them just get destroyed. And time will tell whether this is going to be a big problem. But I can tell you one thing, that from a conservation point of view, we can enclose what we're doing. If we can improve yields, we don't need to devote as much land to farming, and we can put aside land for conservation, for making sure that diversity continues. But I think we've done a hell of a job of destroying biodiversity without GM crops, right?
Yeah.
I mean, do you know how many, the extinction rate is way up there at the moment. No GM crops involved, we've just done it because we're humans and rather stupid very often.
Yeah. Thank you.
Okay.
Thank you for the answer.
Sorry.
Hello, sir. My question is related to nowadays gene editing is being worked for human disorders like sickle cell anemia, thalassemia. So how much success rate is there and what could be the further consequences?
So, so far that's been pretty good. So the treatments for thalassemia have been really good. So what are you concerned about?
Yes, sir. Sickle cell anemia and hemophilia like this.
Right. Well, and so...
It's CRISPR-Cas technology.
Yeah, the experiments have gone very well, the clinical trials have gone very well. I've not seen anything that suggests that's a problem in any way.
Okay.
So are you concerned about a problem, or you want to know what else we can do?
Yeah, I'd like to know about the
So at the moment,
consequences.
anything that has a somatic cell origin, so we can fix somatic cells, and it doesn't seem that it's going to be a problem. The regulatory agencies have pretty much said, yeah, that's okay if you're trying to solve the disease. But germline manipulation is out at the moment, as it should be. It shouldn't be allowed. And when you mess around with somatic cells, you really don't have much of a problem. There's not a big risk. They don't get into the germline very easily. So I think all that is very good. I like it. And I tell you, a lot of hemophiliacs love it too.
Thank you, sir.
Okay.
Hello, sir.
Sorry to interrupt, but we'll take one last question from a student, and then after that, if you have questions, you can either write to him.
Yeah, email.
Or there is also an interaction session in the chemistry department, Pune University, at 2:30. So you can join in there. So one last question.
Okay. Hello, sir. Hello.
Okay.
Yes, sir. My name is Isham Chauhan.
I saw a microphone heading that way, and so I...
No problem, sir. So my name is Isham Chauhan. I'm a first year post-graduation student here in the field of botany. It is one of the most inspirational moments of my life talking to you. And just out of curiosity that we have seen the BT cotton, we have seen the BT mustard. just like that, what is that one, I just want to ask that, what is that one thing which is lacking in the field of science and technology and in the field of GMOs, that we don't have all the crops which we eat are not GMOs? What is that thing like which is lacking behind in the science?
Well, so I mean, I think because of the government regulations and the anti-GMO sentiment, money is missing. Because an awful lot of people have not been able to do the work. You know, the reason I got involved in this, I went to a conference in Ghent in Belgium, in which the originator, Mark van Montague, who figured out what the GM method was, they had a celebration for his 80th birthday, and I sat and listened for a day, listening to European scientists saying how they couldn't talk about their work because Greenpeace came and harassed them. They couldn't get funding because Greenpeace had had political activity and stopped a lot of the funding. They couldn't do the work. They knew what to do, but they couldn't do it. They didn't have the resources. And so we have to make sure that we stop this anti-GMO political movement, remove many of the regulations. It's okay to have some, but the regulations should be the same for traditional crops and for GM crops. There's no reason why they should be different. When we do that, then I think you're going to see there's going to be a swelter of stuff coming along that will be really good, but good for the developing world. The Europeans don't need it. You know, it's the developing world that needs it.
Thank you so much, sir. There is a friend of mine who is back there, and she just want to know that the feeling when you got that Nobel Prize.
Well, so I'll tell you the story. I got a call at 6 in the morning. I'm working on a manuscript. I get a call at 6 in the morning, and my wife is in bed, and there's a phone next to the bed, and I was really pissed off. I said, who is calling at this time in the morning, waking my wife who's going to stop me working? So I go to the phone, I pick it up, and I said, who the hell is this? Do you know what time it is? And the voice at the other end said, this is CNN. Have you seen the news? To which I said, well, why the hell would I be looking at the news at this time of the day? And then the reporter said, well, it's just been announced you've been awarded the Nobel Prize. So it didn't seem quite so bad after that. Okay? No, it's pretty good. I recommend it to everybody.
Thank you, sir.
Thank you very much, Dr. Roberts, for sharing your very interesting journey with us. Dr. Vani would like to present you with a token of our appreciation on behalf of all of us.
Okay.
Thank you.
Thank you. Thank you. Thank you.
Okay.
I need to collect my stuff here.
Yes. So, Professor Avinash Kumbar, Head of the Department of Chemistry, SP Pune University, will propose the vote of thanks. But before that, I just wanted to request the students to stay back after the vote of thanks for a group photograph. We'll take a photograph here in the auditorium first and then outside in the foyer after that. Sorry?
Okay, it's my pleasant duty to propose a vote of thanks. First to Sir Richard Roberts for giving us this fantastic lecture on his path to the Nobel Prize. And as he mentioned that we at the Savitribai Phule Pune University and NCCS are really lucky to have him here visit us. In fact, this visit was planned in March 2020. And he was just about to catch the flight to India, but due to COVID that was cancelled, and we are really lucky that he remembered that he had committed to come to the university, and we were pursuing him through several of our contacts, and he mentioned that this is the time that he could come here, and we grabbed this opportunity, and I'm sure you evidence that this was one of the great lectures that we have listened to, with a lot of motivation, a lot of ideas that he has given us, and also to look back, demonstrated to look back and do a post-mortem and see what things we could have done differently, and I'm sure many students would take this further. I would like to thank NCCS, the team NCCS, Professor Mohan Wani, Professor Shekhar Mande, the team at SPPU, Professor Karbhari Kare, the Vice Chancellor, Professor Sonaune, in giving us support to organize these lectures. I would also like to take this opportunity to thank Professor Mukund Chorghade, who has been really instrumental in connecting with Professor Rich Roberts all throughout these years, 2 years that we have been pursuing with him. Think Pharma from Mumbai, who's also associated with his visit to here. And I would also like to take this opportunity to thank the NCCS team for organizing this, the Department of Chemistry team for organizing this. This lecture has been webcasted, is being webcasted, and I'm sure many students have benefited that, to affiliated colleges as well. And I'm 100% sure there are lots of hits and lots of views that are there, and there will be a lot of questions and lots of emails to you, Rich, for after this visit. And I would like to thank all the audience for coming here. And I'm sure there are a lot of takeaways from this. He also has several tips for us, for an Indian to win the Nobel Prize. And I'm sure the Pune city, the Oxford of the East, would be, at least if not, would be instrumental in that Nobel Prize, which we have been missing for so many years. Thank you, Rich, once again. Thank you all for coming. Thank you very much.
Reminder for students to stay back, please. And if there are students watching in the other seminar hall, please join us here.
नहीं यहाँ से मत बोलो वहाँ जाओ
नाही ना मॅक्सचा हे काय ते इथंच होतं ना नाही ना त्यांच्या लॅपटॉपला गेले काही विचारा पण लॅपटॉप नव्हतं लावलं हां नव्हतं लावलं लॅपटॉप काढून दिलं तेव्हा इथंच ठेवलं होतं अय सागर अरे ते लॅपटॉप निष्काळ दिलाय ना त्याला पिन नाही इथेच ठेवलं